All right, so thank you very much for this chance to bring a bit of microbiology to this forum, which I think it's probably very odd. I am the odd one out. I present from a Mac. I'm talking about microbiology, so really apologize for no seismic lines or anything. But I do hope this will bring some maybe crazy but interesting new ideas for what I understand might not be the immediate future of hydrocarbon exploration, but could be a distant future or an alternative future. But mainly I wanted to show something that is thinking a little bit outside the box. So this is, I'm briefly going to go through one of my projects that is a long-term collaboration between some of us in Newcastle University and University of Calgary, where we asked the question, is biogas production from heavily degraded crude oil a viable option for energy recovery? Can I find the, oh, there it is. So this idea of converting oil to methane is a topic that has been quite recurring in oil microbiology in the last few years, not because of economic reasons at all, but because of the pressure of more sustainable energy production that has been increasing over time. And the idea behind is that we could use any of these waste resources or residual oil or coal, and with the help of biodegradation, produce methane and CO2 with the activity of microbes. And this could be an interesting idea if, for example, we could keep the CO2 down in the reservoir and only extract, facilitate oil extraction, but also increase methane production from that. And this would be great because we could maybe reduce our carbon footprint. And also this could be a way of using the heavy crude oil that we have that is now becoming more the thing to use because we're running out of our light crude oil. Yes, at least that was the thought 10 years ago when we started this. So we started doing this project looking at the very heavy crude oils from Athabasca oil sands and thinking, can we use that to produce methane? And what would it take for that? So this was one of the main questions, and we're also interested in what are the microbes that are involved in this degradation of oil sands, also from the fundamental science perspective of how did the heavy oil deposits were formed in the first place. And we also want to know, is the methane being produced from hydrogen and CO2 or from acetate? And this is important for us, both from the perspective of understanding pathways of hydrocarbon degradation for bioremediation purposes, for formation of heavy oil, but also if we were thinking of doing this as an energy source technology, then we also need to know what exactly are the biochemistry involved and how we can stimulate that. So for that, back in 2005, my colleagues Casey Hubert, Thomas Oldenburg, and Milo van Fustig, they went into these Athabasca oil sands deposits, got some formation water, oil sands samples, and put them in a bottle and made microcosm incubations to look for evidence of this process. And we also looked at the microbial community in the formation water, in the oil sand, and in the microcosms. So what they did, they put them in a bottle, 10 grams of oil sands, that's the only carbon substrate for these microbes. The microbes are coming from both the oil sands and from the formation water. They added a few inorganic nutrients and then incubated for 3,000 days. So that's about 8 years in a bottle. And it's a lot of patience. And they used different electron acceptors. So in 2 of these bottles they had nitrate, 2 of them had sulfate, 2 of them didn't have anything extra, 2 had nitrate and sulfate, and 2 had oxygen in this sealed atmosphere. And they observed that all the microcosms that didn't have nitrate produced methane at some point at different levels. So if you look a bit closer at time points, there are not that many, but you see a production of methane in all of these bottles at different levels, especially in the first 4 years and a bit less in the next 4 years. And I jumped in about day 3000 and started doing some DNA analysis to look at the microbial community that developed in these experiments. And so very briefly, essentially what I did, I sequenced a particular gene that allows us to identify these organisms. and looks for key ones. So, for example, obviously methanogens, which were the ones we were most interested in. So we saw that they were originally present in the oil sand itself and the formation water, but they were enriched. So the relative abundance of these methanogens increased over time in the microcosm that produce methane, which is expected. The nice extra bit of information that we got is that this, the most abundant methanogen that we got in all these experiments belongs to this, very familiar to you, I'm sure, the methanosacina family. And this is important because they're usually known for doing methane production from acetate. However, when we look closer into the DNA, we saw that this particular enriched methanogen does not use acetate. It might actually use hydrogen and CO2, but it uses different compounds like methanol, monoethylamine, and so on and so forth. So this gives us a hint that it's actually this process that is going on, hydrogen and CO2 being used for methane production or methanol and other compounds. When we looked at the ones, the syntrophic bacteria, so bacteria that help getting down to this point, so they start by degrading the larger compounds of oil into something smaller, we saw what we call the usual suspects, Syntrophobacterialis. They're very much found in oil-contaminated environments and associated to these methanogens. But we also found a few other ones that I particularly find it quite interesting, and one of them being sulfur reducers. So I am a big fan of sulfur reducers. I worked with them since my PhD. They're not very popular in the oil microbiology because they cause souring in offshore reservoirs due to the production of hydrogen sulfide from sulfate. However, the ones we find here, they are found in both the conditions that we had with sulfate and the ones without sulfate. So in fact, it looks like they're not doing sulfur reduction. In this case, they're just doing fermentation. So it shows us they are able to do a lot more things over there. So this was interesting, but my favorite are the geobacteria that seem to be, so this is the orange bar here. They were enriched in all of the methanogenic microcosms. And the reason why I get excited about it is because of the crazy possibility that they might be doing a direct interspecies electron transfer. So what does that mean? That means that it has been found recently, so just in 2014, that some of these bacteria can transfer electrons directly to a methanogen. So these are not them, these are different species, but I guess it will do. They make this conductive pili that connects different cells, and they're able to just conduct electricity from degrading a carbon source and then just transferring the electrons to the methanogen, and the methanogen can grow based on that. That is pretty cool, and I'll tell you why later. We can't really say that this is definitely happening in our oil sands experiments, but it opens some possibilities for crazy ideas. So going back quickly, we did see possibly this side is happening more than this pathway. We saw a bunch of different bacteria being involved in the upper levels of hydrocarbon degradation with this oil sands bitumen. However, there's a big problem, which is the amount of methane that was really produced, which is very, very low. So the maximum that we got here was about 40,000 ppm after almost 4 years of incubation. That is really not going to be very helpful if you want to think of this as an industrial possibility. And when we looked at which oil sand compounds were really degraded, after 8 years in these anaerobic microcosms, we did gas chromatography and spectrometry to see if we can find any decrease of any compound, and we found none. We then went full on and did Fourier transform ion, cyclotron resonance mass spectrometry, so ultra high resolution mass spectrometry. And again, we found nothing. There's no change on anything specifically. So we did see production of a few strange compounds, but we think that these are probably just cell biochemistry related compounds. So where does the methane really come from in those cases? We know it's biogenic because when we look at the signature of delta 13C, it became depleted in delta TC, so the methane, it's an evidence of enzymatic fractionation. And so one possible explanation what is happening in these experiments is that there's a very slow, unspecific degradation of various oil compounds at the same time. So it's very difficult to characterize the biochemistry that is happening there. And when we compare the rate of this methane production in these experiments, it's 10 to more than 1,000 times lower than in other experiments where we see biodegradation of waste oil treatment tank or lighter crude oil and so on. However, the microbial potential is there to degrade a lot of different things because, for example, when we get the same oil sands and add benzoate to it, then the methane production from benzoate happens at almost like the same order of magnitude and rate as, for example, when you use river Tyne sediment, which has also a variety of microbes that are able to degrade several different organic compounds. So it's really the problem, or the bottleneck is on the carbon composition, but the microbial potential is there. So in conclusion, we did see this evidence for methane production, and we saw enrichment of methanogenic archaea and several other bacterial syntrophs that are participating in this process. And we saw that the sulfur reducers, the villains in offshore production, may actually be doing fermentation instead of sulfur reduction in these oil sands. But we saw no degradation of specific oil compounds using two different techniques. So when we come back to that original idea, can oil sands be used for oil to methane production? Well, it's very impractical, but maybe, just maybe, with some more elaborate bioengineering, we might be able to do something like that. And that's where I come back to this possibility. If we could take the methanogen out of the equation and just harness the electrons that these bacteria can produce from degrading oil compounds, then maybe we could use that for electricity generation. This is probably going to run into several problems, including resistance, for example, to get electrons from here to there. But it is a potentially interesting idea. Also, if we think of other ways, instead of getting electrons directly, if we use electron shuttles. So this is an idea from Max Charles and Steve Larter in Canada, and they are currently working on testing that hypothesis. And one idea is, for example, we run into microbiological problems in the offshore, a lot of it because of souring, so hydrogen sulfide production. What if we would actually stimulate production of sulfide and then collect the sulfide up here, oxidize it, and generate electricity from there, and circulate the sulfate back? One massive problem with that could be that sulfide is still corrosive, it's still toxic, it's still explosive. But if we can think of other electron shuttles that we could use in this process, that we can just introduce an oxidized version here, get the reduced version, and then keep all the hydrocarbons down in the reservoir, that would be a zero carbon electricity production, which could be an attractive or an interesting out-of-the-box idea for a alternative future hydrocarbon exploration. I don't think that this will solve all the energy demand of the world at all, but I think it's just some food for thought for what we can do when we think of changing paradigms in the science. That's it. I thank a bunch of collaborators over time, and Microbiology Society for giving me a travel grant to be here, and thank you for your patience to listen to microbiology. Thanks.
Thank you for a very interesting talk. Have you got any questions from the floor?
Did you try in any of the experiments to mix your samples? I'm just curious, thinking about, I mean, I'm a geologist, the oxidation of organic matter certainly is greatly enhanced when you mix the sediment. So I just wondered if you've looked into that at all.
These ones, they were in a rotary shaker for maybe, I think, the first year or two. But the other problem that we have when it depends on microbiology degradation is that often they need to be in physical contact, so you don't want to shake it too much, because, you know, especially for example, if those nanowires are one of the way that things are being produced, you actually need them to be relatively, yeah, quiet. But yes. Mm-hmm.
Could this be used in depleted fields? So is there a minimum concentration of carbon required for it?
I might not be entirely familiar with the term depleted field, I'm sorry.
So old fields that have already been producing for 30
Oh,
years.
yes, yes.
Is there a minimum carbon concentration required for this reaction to occur?
No, not at all. I mean, I would actually suggest, or one of the suggestions we get from this is that it's probably not great for oil sands, because they are really, really recalcitrant, difficult material to use by the microbes. But if you have residual lighter oil, then this becomes, you see, the rates of methane production can go up orders of magnitude if you have a better oil. So yes, this would be a better way of using it for sure. Mm-hmm.
Any more questions? Okay, just one more.
Do you
Yeah.
have plans to move from the lab to industry to scale up?
This is, so not me, I'm more focused on the souring project, so this is a side project. But Mark Strauss and Steve Latta, they are currently working on testing the hypothesis in a small lab scale, but in maybe 2 years or so, make it larger scale.